Patellar tap reflex again. We talked about it yesterday, but I want to lay it out today in the same fashion where we talked about it last time, where we talked about the unilateral effects, that is on one side. I want to lay this out as a bilateral structure today, and we'll talk about the information coming in from both the right and the left legs. And we're going to deliver this somatosensory information then first to that circuit we talked about yesterday. But for us to know where we are in space, to be aware of what's going on around us, to be able to touch something and feel it, that information can't simply be relegated to these reflex arcs. We have to take that information up into the brain, and we're going to follow the pathway of information going from the periphery into the central nervous system and up to the somatosensory regions in the somatosensory cortex and the cerebrum. And that's going to be this process of following the pathway. So this fundamentally is delivering somatosensory information. And what do we mean by somatosensory information? Well, somatosensory information comes in a number of forms. One of the major forms is what we call proprioception. Proprioception is telling us where our bodies are in space, and we do this all of the time. And those stretch receptors that we talked about here for the patellar tendon tap reflex, those are really important in telling us how much tension we have in all of our muscles, and thereby telling us to some extent where our muscles are in space. It's the proprioception which allows you to close your eyes, take your finger, and touch your nose. It's that because you know where your hand is in space, you don't have to see it. You know where your nose is in space, and so you can touch your nose with your finger, with your hand. That's proprioception. But in addition, there's a lot of other sensory information coming in from the body. That is, of course, as you're sitting there writing, you can feel the finger, you can feel your fingers, you can feel the touch between your pen and the paper. All of that is going to be something called fine or discriminative touch. Discriminative touch is this ability to figure out where things are in space simply by touching them. And that's information, the proprioception and fine touch, is going to be carried into the central nervous system on primary sensory afferents, synapse onto second order neuron to third order neuron, and it's going to ascend the cord in something known as the dorsal column medial omniscial pathway. It's going to go it will go from the peripheral nervous system into the central nervous system, ascend the cord, cross the midline in the brainstem, and find its way to the somatosensory cortex through this dorsal column, sometimes known as the posterior column medial omniscial pathway. So hold on to the idea of DCML or PCML, proprioception and fine touch. The other character we're going to talk about will be carrying the different kinds of feelings we're aware of. That is, think about pain or pressure or crude touch and temperature. Those are all coming up through this alternative pathway called the spinothalamic tract. So we'll keep separate the idea that we have proprioception and fine touch entering the central nervous system and going to the brain on one pathway called the DCML or PCML, and pain, pressure, and temperature coming through another pathway called the spinothalamic tract. And we'll follow these two. For both cases, the rules for both cases are very similar. Whether we're talking about the PCML or whether we're talking about the spinothalamic tract, they are all composed of a three-cell chain. A primary sensory afferent synapsing onto a second-order neuron in the central nervous system. That second-order neuron then crosses the midline, and then once it crosses the midline, it will then synapse onto a third-order neuron. The third-order neuron is always located in the thalamus. The thalamus is a common sensory relay site. It doesn't matter whether you're talking about auditory information or visual information. All sensory information goes through the thalamus, and that's true here as well. So first-order neurons in the periphery always stays on the same side of the body. The second-order neuron always crosses the midline. The third-order neuron has its cell body in the thalamus and extends a process into the somatosensory cortex, where in the cerebrum it forms synapses. So we'll follow this pathway. And then, as a kind of exercise, a kind of preliminary exercise in neuroanatomy and sort of using neuroanatomy to diagnose problems, and we're going to walk through something known as Brown-Séquard syndrome. Brown-Séquard syndrome is a case where the spinal cord is severed just to the midline, not completely through, but just to the midline. And it's a useful exercise, not so much because of the Brown-Séquard, but because if you can follow your way through the Brown-Séquard syndrome and understand why the effects that Brown-Séquard syndrome shows, if you can see that, then you understand the pathways where this information is being carried. More importantly, this idea of Brown-Séquard is what neurologists do all the time. Neurologists are interested in what's broken in a nervous system. And almost always neurologists are outstanding neuroanatomists, because what they're doing is they're looking at a deficit and then trying to puzzle through where those deficits originated. And they do that largely by understanding the neuroanatomy. And this is a very fundamental case of using neuroanatomy to understand what's broken. And we'll walk our way through that. Once we have done all of this and we've delivered the information into the cerebrum, we'll talk a little bit about the overall structure of the brain. And we'll do this in the process of talking the brainstem, the thalamus, and the cerebrum. And we'll walk our way through this, at least to some extent, by following the pathways of the spinal thalamic tract and following the pathways of the dorsal column system. But the final target of this three-cell chain in the somatosensory system is to find its way to the cortex. Now, we can't spend a whole lot of time on brain anatomy here. That's relegated to BMS 345 or BS 545. But I want to spend a little bit of time just introducing you to some fundamental ideas of the cerebrum and the cerebral cortex. When we talk about the cerebrum, the cerebrum is the part of the brain when we think about as being the brain. When you envision the brain and you see that folded structure, that's the cerebrum. So a lot more to the brain than the cerebrum. But the cerebrum is where a lot of these sensory pathways will in fact terminate. The sensory pathways that we're going to talk about terminating are the somatosensory pathways. And they're going to terminate in a particular region of the brain. And this again is going to bring up the idea, the fundamental concept that function is compartmentalized in the brain, that different parts of the brain do different things. There's a big argument about how far down this compartmentalization goes, but certainly there are certain functions that are relegated to certain parts of the brain. And we know that the somatosensory information is coming into the part of the brain called the somatosensory cortex. And it's located in a particular region called the post-central gyrus. And now we'll introduce us to a little bit of anatomy, talking about the various landmarks in the brain. And again, if you take a neuroanatomy course, you'll talk about these landmarks and you'll talk about the various regions in terms of the various landmarks. But here, what we're going to do is we're simply going to walk through first the idea of the somatosensory cortex, how those three-cell chains terminate in the somatosensory cortex. And then we're going to talk about how the somatosensory system is found on the post-central gyrus, but that those third-order neurons don't make synapses randomly in the somatosensory cortex. There's a highly ordered fashion in which those synapses are formed. And that highly ordered fashion can turn out to be a map of the body plan. And so we'll see that there's a particular region of the somatosensory cortex that is dedicated to sensory information coming from the face. Another area dedicated to sensory information coming from the hand. And this is always mapped out in the same way. Anyone in this room, if we look at the map, is going to be pretty similar. And so we'll walk our way through that and what it means. Now, that's going to bring us to the idea we've brought all the somatosensory information in, but remember from the three-box model, we have sensory in, some kind of integration, and motor out. Again, we don't have time to to talk much about this parietal association areas, but I'll spend a moment on it because what happens in the parietal association areas is that we have sensory information coming in from any number of modalities. And what we mean by that is somatosensory information will be combined with visual information, will be combined with auditory information. It's in the parietal association areas that we begin to make sense of the world around us by bringing all the sensory information into one place. Then that sensory information, which is compiled in the parietal association areas, will be then sent forward to at least to some extent, the forebrain, and in the forebrain, this information then will be analyzed and a decision will be made on what kind of movements to make. So again, this is back to the three-box model. Sensory in, we'll have a lot of the analysis going on in the parietal area, then you have to decide to do something. And we're going to simplify this a lot. There's several areas involved with this. But the main one we're going to talk about is something known as the motor cortex. The motor cortex is found on the precentral gyrus. So it's just one bump over from the postcentral gyrus where we find somatosensory system, but this is going to be motor. We're going to find that in the motor cortex there are specialized neurons called upper motor neurons. Sometimes you'll see these referred to as BET cells. They're gigantic cells. And they have processes that go all the way from the somatosensory cortex down to the spinal cord. And in humans, there's a monosynaptic pathway that goes from the upper motor neuron to the lower motor neuron. And the upper motor neuron will drive the lower motor neuron, as we've alluded to up to now. That lower motor neuron will then cause synapses in voluntary muscle to cause muscle contraction. So what we're going to do today is we're going to work our way through the somatosensory system, work our way into the brain, look a little bit at the association cortices, and then have the descending information come back out to muscle. Before we do that, let me remind you of what we're looking at. And then I'll put some of the crude drawings on the board again. So this first image is a prettier picture of the spinal cord than I'm capable of drawing. And it shows the spinal cord a little bit into three dimensions, and it summarizes many of the things that we talked about yesterday. And if you can envision this in your mind's eye, you have a pretty good sense of what the cross-section of a spinal cord would look like. And in addition to the cross-sectional arrangement of the spinal cord in this image, we also see the rudiments of the longitudinal arrangement. What I mean by that is if we look here, we see these three pairs of spinal nerves. Remember, all along the spinal cord, there are 31 pairs of these, and we're simply showing 3 of them here. But now, out here in the lateral portions, those are actually the spinal nerves. The spinal nerves are a combination of lower motor neurons exiting the spinal cord through the ventral through the ventral root, and a combination of sensory neurons, primary sensory afferent neurons, carrying information toward the central nervous system. The neuronal cell bodies of all of the somatosensory neurons are located here in the dorsal root ganglia. So there are 31 pair of dorsal root ganglia. Each of those segments will be responsible for a particular part of the body. And all of that's mapped out. We know where each of these dorsal root ganglia sensory neurons are coming from. And so all of this, everything outside this round, more or less ovalish spinal cord is in the peripheral nervous system. Everything inside is the central nervous system. And then within the spinal cord itself, there's this sort of dark brown region that looks like a butterfly. That's going to be called the gray matter. And then outside of that will be this sort of lighter brownish, yellowish structure, which will be known as the white matter. But again, this is a repeating pattern. So you have 31 repeats, 31 segments along the length of the spinal cord. What you see on the right, what you see on the right side, you see on the left. So the arrangement of this is as though I'm standing here facing you, and my back is dorsal, my belly is ventral, also known as posterior and anterior. And when you look at this then, this is going to be the dorsal horn, because whenever you see horn, remember we're talking gray matter in the central nervous system. Here will be the ventral horn, and this is gray matter in the central nervous, in the spinal cord. And this ventral horn will be will contain the cell bodies, the lower motor neurons. And what we talked about yesterday was the reflex arc. Let's do this one first. So this is the anatomy of the reflex arc, showing the primary sensory afferent. This is the ending. The neuronal cell body for the primary sensory afferent will always reside in the dorsal root ganglion. The process of this primary sensory afferent then will extend into the central nervous system. And particularly, it's going to extend into the central nervous system, into the gray matter. And in the gray matter, this primary sensory afferent will have a synapse directly onto this green lower motor neuron. So this is a monosynaptic pathway. Also, the branch of this primary sensory afferent is going to synapse onto an inhibitory interneuron. So when the generator potential is set up here at the input region by tugging on this input region, then the generator potential will initiate action potentials or propagate all along the axon over the cell body and into the central nervous system. And at the output region here, 4, neurotransmitter in the form of glutamate will be released onto the lower motor neuron. The neurotransmitter will set up a excitatory postsynaptic potential, a really big one that almost every time will fire action potentials in this lower motor neuron. Those action potentials now will propagate to the output region of the lower motor neuron, synapsing onto skeletal muscle of the quadriceps muscle. And what will happen then is by stretching this input region and establishing action potentials that release neurotransmitter onto the lower motor neuron, this monosynaptic pathway will result by stretching the tendon, pulling on the input region, firing action potentials to release neurotransmitter to fire action potentials in the lower motor neuron to cause the quadriceps muscle to contract. But we said that there was also this idea of reciprocal inhibition, that when we excited one side, we inhibited the other, and that was because of the antagonistic nature of skeletal muscle in mammals. And what we see then is the antagonistic muscle to the quadriceps is the hamstrings. So whenever we fire action potentials in this lavender axon, this lavender lower motor neuron will fire action potentials, release neurotransmitter, and cause the hamstrings muscle to contract. So when we contract the quadriceps, we want to have the hamstrings muscle turned off. Alternately, when we contract the hamstrings muscle, we want to have the quadriceps muscle turned off. Now, the pathway that we show here is turning on the quadriceps muscle and turning off the hamstrings muscle. That turning off occurs here. We turn off the lower motor neuron innervating the hamstring at this inhibitory synapse. So again, almost simultaneously, when action potentials arrive at the output region of the primary sensory afferent and release glutamate onto the lower motor neuron, that primary sensory afferent is releasing glutamate onto this inhibitory interneuron. It's an excitatory neurotransmitter, so it's generating excitatory postsynaptic potentials that summate and generate an action potential in this inhibitory interneuron. Those action potentials will now arrive at the output region, and they'll release a neurotransmitter. What neurotransmitter will that be at this site? GABA or glycine. What kind of channels are they going to open? Is that neurotransmitter going to open in this neuron? Chloride. That's going to inhibit or turn off this lower motor neuron. So now we have this reciprocal inhibition where simultaneously when we cause excitation to the quadriceps, we cause inhibition through inhibiting the lower motor neuron to the hamstring muscle. Now, one of the things that's shown in here, but I need to point out, is that there is no peripheral inhibition. In the voluntary muscle system, there's no peripheral inhibition. All inhibition occurs here in the central nervous system. Then this next slide shows the electrical events in that pathway. It starts out here with the muscle spindle. And just as we talked about the stretch receptor neuron at the tip of our finger, when we pull on that muscle spindle, when we increase the tension or stretch on the muscle spindle, what we're going to do is we're going to generate a graded receptor potential here in the muscle spindle. That will initiate an action potential. The action potential, or probably a series of action potentials, will propagate along the sensory neuron. When the action potential arrives at the output region of the sensory neuron, we then activate the voltage-gated calcium channels and go through the whole snare story, releasing excitatory neurotransmitter glutamate onto the lower motor neuron. This sets up excitatory postsynaptic potentials in the input region of the lower motor neuron. Now, the innervation pattern of the primary sensory afferent onto the lower motor neuron is so strong, there's so many synapses and so much neurotransmitters released that it's almost invariably a case where this graded synaptic potential will be large enough to bring the membrane at the trigger zone above threshold to fire an action potential or series of action potentials that now propagate into the periphery on the lower motor neuron out to the muscle, where the lower motor neuron releases neurotransmitter. We'll see a little later, it's acetylcholine, to cause an EPSP in the muscle, followed by an action potential in the muscle that causes contraction. That's the pathway. So what we're seeing here is the synaptic relays, and talking about the neurotransmitters that are released at each of those relays. And what we're talking about in this slide is the anatomy of those neurons. And if you study these two together, and these are both in the ebook, if you study these two together, you'll be able to sort out where the neurotransmitter is being released, where excitation is going on, where we have graded potentials, and where we have action potentials. Separating, again, the importance of action potentials from graded potentials. This next slide is one which we're really going to talk about today. And so as we go through what I talk about, what I put on the board, remember that we can divide the spinal cord into different regions. The major differential here is going to be this butterfly region in the middle that we call the gray matter. Whenever you see gray matter, you immediately think synaptic inputs. Those are the input and output regions. So gray matter includes sites where we make synapses. Outside the gray matter is what we're showing here in yellow. That's what's called white matter. The white matter gets its name because it's sort of a creamy white color. And it's a creamy white color because of the fat that is in the spiral wrappings of the oligodendrocytes that have laid down the myelin. So when we talk about white matter, what we're really talking about is myelin. And all of these regions out here are myelinated axons. And those are the long distance communicating portions of the neurons. We've gone through that a number of times with input regions, conductal regions, and output regions, and these will be the conductal regions. If we drill down a little farther into the white matter, even in the gray matter, we find that there are subregions within these structures. And the subregions we're going to talk about today will be up here. These regions are known as the dorsal columns. These dorsal columns will have processes of the primary sensory afferents. So we're going to draw the reflex arc initially, but recognize that these primary sensory afferents, although they will send processes into the central nervous system, they also have a branch that enters the dorsal columns. And that branch then will ascend the cord, and that will be part of the posterior or dorsal column system, and we'll talk about that. The other major bundle here that we'll talk about is the spinothalamic branch. In the spinothalamic branch, we'll see that neurons whose cell bodies are in the spinal cord, spinal, will have synapses in the thalamus, and that will be the spinothalamic branch. And if you remember what I said in the introduction, the spinothalamic branch here is going to be carrying information about pressure and temperature and pain, whereas the dorsal column system will be carrying information about fine touch and about proprioception. And we'll walk our way through that. And then in red, we'll see their descending pathways. So part of this figure 2 is to distinguish in blue that which is carrying information toward the brain and the ascending pathways. And those regions then have their neuronal cell bodies in the brain and will be carrying information in a descending fashion coming down the cord, and that will be primarily motor. But let's, again, so let's start here again with a drawing a cross-section of the spinal cord, and I'll try to make it big enough because we're going to continue writing on this for some time today. And to begin, that's intended to be the spinal cord. We're going to say just that this is right and this is left. And so it's as though my head were behind the board with my feet on on the table, just as we did yesterday. Then within this, there is this bilaterally symmetrical structure called the gray matter. This is all gray matter. If you look at many figures, they'll show this round structure in the middle. This is said to be the central canal. It's actually part of the ventricular system of the brain. There's cerebrospinal fluid in it. We're not going to spend any more time with it. But from this point, the first thing I want you to recognize, and I'll put this in purple, is that we can divide the spinal cord in half, such that we have a right half and a left half. So we're bilaterally symmetrical. We can also divide it along the midline here, and pretty much say anything above this midline is posterior, sometimes known as dorsal, and anything below the midline is going to be ventral or anterior. And then we said yesterday that there was a kind of interface, a kind of separation, which we denoted by a red dotted line. And the red dotted line is meant to demarcate the boundary between the peripheral nervous system and the central nervous system. So everything inside the red dotted line is central. Everything outside the red dotted line is peripheral. We then said that up here was a short segment of dorsal root, which then expanded into this sort of bulbous structure, which we said was the dorsal root ganglion, sometimes known as the posterior root ganglion. And what we see on one side, we see on the other. You see this short section of dorsal, sometimes known as posterior root, and this dorsal root ganglion. Then on the ventral side, or the anterior side, we saw coming out from the spinal cord. I'll show this. Let me show it in another color. Let me show it in brown. We had this structure known as the ventral, also known as the anterior root. Again, when you see this term root, it's in the periphery. So this is going to be on the red dotted line. And with my sort of primitive color scheme here, what I'm going to try to do is show that the ventral and dorsal roots actually fuse and give rise to this common structure out here known as the spinal nerve. So the spinal nerve is fundamentally a fusion of the dorsal root as it exits the dorsal root ganglion and the ventral root. So they'll combine. And what that means then, if we follow the neurons, is that there will be neurons down here in the ventral horn. In the ventral anterior horn, there will be these neurons which we're going to describe as lower motor neurons. And these lower motor neurons send processes out into the periphery. This fundamentally is what we were talking about yesterday. And out here, we can imagine an individual's right leg. And setting up the patellar tendon wrap cap reflex. Now, as I draw this, recognize that there will be a whole series of muscles all throughout the body, and all of them will contain stretch receptors, and all of them, if you know where to stretch the tendon, all of them will generate this tendon tap reflex. But the one we emphasized was the one that was predicated on the link between the quadriceps and the patella, and its anchoring point between the quadriceps and the bones of the lower limb, the fibula and tibia. Now, the image that we showed just a moment ago on the slide showed this input region of the primary sensory afferent. And if we follow the axon of this, I'm going to show it as a line, but remember this is going to be a proper cylinder cytoplasm surrounded by plasma membrane. This is going to have a process that goes into the nerve and then has its neuronal cell body here in the dorsal root ganglion, and then continues a process into the spinal cord. This primary sensory afferent. The primary sensory afferent is carrying action potentials from the periphery into the gray matter of the spinal cord, and will come down here and make a monosynaptic pathway. It synapses directly onto this lower motor neuron. Now, I should say here, truth in advertising, that this is an unusual pattern. It's only these tendon tap reflexes which have this monosynaptic pathway. If you think of the pathway where you touch your hand on a hot stove, that's a very similar reflex. It doesn't have to go to the brain to get you to pull your hand back. But that reflex actually has a primary sensory afferent that's sensitive to heat and pain. That neuron will make a synapse onto an interneuron, and the interneuron then in turn synapses onto the lower motor neuron. So it's a little bit slower by a couple of milliseconds, but not a whole lot. This is a highly selective pathway. And where we use this pathway is not just to get whacked on the knee at the doctor's office. We use this all the time as monitoring the stretch in our muscles. And the most common place you can think about using it, and we've all done this, you walk along the sidewalk and you catch your toe in the crack of the sidewalk, and your right foot stays behind a little bit, just for a little bit, and so you put a little bit extra tension on that. There's a reflex arc here, which now will immediately cause the tension in the quadriceps to increase the rate of extension, so you put your step forward, and it also will help you plant your left foot. So if you've ever done this and thought about what you'd just done, although you usually try to show you didn't trip over the crack in the sidewalk, what really happens is your back foot gets caught, and you'll quickly move your weight to the left foot and then quickly bring your right foot forward. That's largely the stretch receptor reflex. And so that there are also going to be synapses that go to the other side. We're not going to talk about those, not in this course. But what we have is a monosynaptic pathway. We fire the action potentials, release a neurotransmitter, and we activate the lower motor neuron, which then synapses onto that quadriceps muscle. But at the same time, remember, over here is the antagonistic muscle of the hamstring. It too receives synaptic input from a lower motor neuron. Often a lower motor neuron is in the same neighborhood in the ventral horn. But now, you'll remember from the slide, there's a branch of this primary sensory afferent. Let me make it short. There's a branch of the primary sensory afferent, and that Y indicates that it's going to synapse onto an interneuron, which now synapses onto this lower motor neuron. And this interneuron is an inhibitory interneuron. Now, quickly, what I want to do here is I want to show to you that this is a truly bilateral system. So everything that we see on the right can be repeated on the left. I won't draw on all the detail, but recognize that this is the quadriceps. And the quadricep on this left side will contain the stretch receptor neurons, the endings of the stretch receptor neurons in those we call muscle spindles. And this neuron will then send a process into the dorsal root ganglion. So this is all one neuron. And this other extension of the neuron goes into the central nervous system, goes into the gray matter of the spinal cord, goes down into the ventral horn. So the cell body's in the dorsal ganglion. It extends the short segment into the dorsal root. It then enters the gray matter, here known as the, we do it this way, dorsal horn. Synapses onto a lower motor neuron in the ventral horn. The neuron exits the cord in the structure known as the ventral root. This axon of the motor neuron then innervates the quadriceps muscle. And the pattern is the same on the left and the right. The ventral root and the dorsal root effectively unite and form, well, it's all short, and form this structure known as the spinal nerve. Everything we see on this side, we see on this side. So there is also a lower motor neuron. And I should add, truth in advertising, there will be many, many of these. We're not talking about one for one. There'll be hundreds of them. And this is a lower motor neuron, extends its process out. In this case, its target is going to be the hamstrings. And again, too, there will be a branch of the primary sensory afferent, which synapses onto the green inhibitory interneuron. So that whenever we excite the quadriceps muscle, we almost simultaneously, through this inhibitory interneuron, inhibit the lower motor neuron going to the hamstrings muscle. So this is mainly to show what we see on the right, we see on the left. And at the close of yesterday's discussion, we pointed out that if we would cut the spinal cord at some point above this, more toward the brain, that this circuit would remain intact. But the problem that would arise is that we couldn't deliver information from the muscle or the periphery to the brain. So what we want to think about now are the pathways that are going to be delivering the information to the brain. So any questions here? Yes?
So you have the dorsal horn inside, but then the ventral horn on the other. Does that mean that there's the same sides?
So these pieces in the central nerve, those are horns. So these will both be ventral horns. But sometimes, because of the nomenclature, in animals it's called dorsal and ventral. In humans it's called posterior and anterior. So I sort of use those interchangeably. So if you see anterior, I'm really talking about ventral. If you see ventral, I really mean anterior. And in the same fashion, dorsal is often known as posterior. So when I say dorsal, I often mean posterior and posterior dorsal. They're the same thing. It's just the nomenclature is different in animals than in humans. And I always worry I mislabel them, but I think, is everything okay here? Okay. Now, this individual neuron, this primary sensory afferent with a single conductal region out here in the periphery, branches here to innervate the inhibitory interneuron, but it also branches here and enters this region of the spinal cord, which I'll hash in orange, known as the dorsal or posterior columns. With the upper panel. The dorsal columns are white matter tracts. Now we need a definition here. Whenever you see the term tract, what you're talking about is a bundle of conductile processes in the central nervous system. The counterpart, of course, in the peripheral nervous system is a nerve. So a nerve is a bundle of conductal processes in the periphery, whereas a tract is a bundle of conductal processes in the central nervous system. The dorsal columns are a great example of a tract because they contain all of these primary sensory afferents that are carrying information about fine touch and proprioception. So that's the pathway we're going to follow first. We're going to follow a pathway of these neurons, these primary sensory afferents that are carrying information about proprioception and touch. We're going to follow them up the spinal cord. So any questions before we leave the cross-section? Yes?
So you're showing one cross-section of one of the 31 segments. Does the, do the primary sensory afferents and all the connections go up all of the, like, because you're showing you just go through.
Oh, okay, so the question here is what we're showing is one segment. And we could look at, and if this is coming from the leg, it's going to be somewhere in the lumbar region. And we could look at any of the segments more cranial or toward the tail caudate. And we would see the same pattern. We would see that these neurons come in. We're not going to talk about it here, but if you take another anatomy course, you'll find that those primary sensory afferents that come in from the legs are going to be in one bundle. And they'll all be mapped out so that ones from the toes will be in a region, one from the calf will be in a region, one from the thigh will all be in pretty much the same region. And as we go from segment to segment, these guys will join one to the next, and they'll all maintain this, what they call somatotopy of where they came from. But you can imagine these bundles of wires, each coming in, and they are all residing in the same region. They're not all mixed up. They're basically one side by side. Does that answer it? Okay. So this pattern basically repeats itself 31 times. And what I'm going to do up above is try and draw that in the longitudinal view that we started with yesterday, where this is the ice cream cone diagram. Make it a little bigger. And if this is the spinal cord, there will be these 31 pair of spinal nerves. This then, let me make this a little, this then expands into a region known as the brainstem. And above the brainstem is going to be a region known as the thalamus. And above the thalamus, in blue here, is going to be a region called the somatosensory cortex. And what we're going to do here is we're going to begin following this three-cell chain, which is going to deliver information from the periphery all the way to the somatosensory cortex. Now, we're going to start someplace down here in the lumbar region. We'll just say this is lumbar. There's going to be the dorsal root ganglion. The neuronal cell body that we're talking about here is the one we have in cross-section above. This neuronal cell body will have a process in the periphery that goes to the quadriceps muscle. So we're just repeating what we have above in this longitudinal view. The neuron has an extension to the central nervous system, which we described above, going into the gray matter. And if we would follow it, it would grow into the gray matter too. But what we're interested in now is following the branch of this neuron that enters the dorsal columns, the posterior columns, and this neuron will ascend the cord. You could stand on this neuron in the muscle and walk on it step by step all the way into the dorsal root ganglion. Walk into the spinal cord, and then you'd reach a branch point. You reach a fork in the road. And if you took one turn, you'd go down into the gray matter and the ventral horn. If you took the other turn, you would enter the dorsal columns. Just emphasizing this is one neuron with its same plasma membrane all along this region. This one neuron, though, finds its ending here in a site called the dorsal or posterior column nucleus. Again, this term warrants a definition. When you see this term nucleus, a nucleus is a cluster of neuronal cell bodies in the central nervous system. So in this case, this nucleus is going to be a cluster of neuronal cell bodies. There may be thousands of these. And this green dot here now is the cell body of the second order. Neuron. Primary sensory afferent sends a process into the dorsal columns, ascends the cord, and in the nucleus, and because these are cell bodies, this is effectively input regions. Input regions for the second order neurons and output regions for primary sensory afferents. Remember, primary sensory afferents never cross the midline. So that obeys this rule. This is the midline. This divides the spinal cord in the brain into right and left side. So if we say this is right, and this is left, what we find is this second-order neuron whose cell body is in the dorsal column nucleus. This neuron sends a process out, and it crosses over the midline. This structure is known by and large as the medial lemniscus. This neuron then crosses over the midline in the brainstem and ascends the cord to a point where it reaches another nucleus. This is known as the this is known as the thalamic relay nucleus. Again, it's a cluster of neuronal cell bodies in the central nervous system. It's a site where the output regions of the second-order neuron synapse onto the input regions of the third-order neuron. The third-order neuron, now whose cell body or input region is located in the thalamic relay nucleus, this neuron sends a process out to enter the somatosensory cortex. So before we take a break, let's just look at this in some detail. Primary sensory afferent 1. Second-order neuron crosses over the midline. Synapses onto third-order neuron. Third-order neuron now extends to the somatosensory cortex. And so we now have delivered information about proprioception through this 3-cell chain to the somatosensory cortex. And at each site where we have a synapse, there's a possibility of some modulation of that information. But that's the 3-cell pathway. Let's take a break, and then when we come back, we'll follow a similar but slightly different pathway for information about pain, pressure, and temperature.